Welcome to Digestion, Absorption, and Use of Biomolecules in the Human Body, a virtual field trip. Food contains biomolecule polymers and monomers. One carbohydrate polymer present in a sports nutrition bar, for example, is a carbohydrate polymer amylose. Let's examine what happens to this molecule as it travels through the human body. Digestion of carbohydrates in the foods we eat begins in the mouth. The enzyme amylase breaks apart the carbohydrate polymer into smaller polymer and monomer units. Let's watch. Here we see the large carbohydrate polymer amylose. As it enters the mouth, it'll encounter the enzyme amylase. Watch as the amylase enzyme breaks apart the carbohydrate polymer into smaller units. When we swallow food, the smaller carbohydrate polymers and monomers travel down to the esophagus into the stomach, where digestion can continue to occur. The enzyme pepsin and hydrochloric acid are present in the stomach. Chemical digestion of carbohydrates does not continue to occur in the stomach because pepsin breaks down proteins. As you can see here, amylose is not able to fit into the active site of this pepsin enzyme. Therefore, it's not able to be broken down further into smaller parts. After visiting the stomach, the small carbohydrate polymers and monomers reach the small intestine. Here they encounter the enzymes amylase, trypsin, and lipase found in biosalts, which break down carbohydrates, proteins, and fats respectively. Let's examine what happens here. As you can see, the smaller carbohydrate monomer fits into the enzyme amylase. Therefore, it's able to be broken down into even smaller parts, including monomer units. However, the amylose does not fit into the active site of trypsin or lipase. Therefore, neither enzyme is able to break down the carbohydrate further. The carbohydrate monomers are now small enough to move across the cell membrane of cells lining the small intestine and enter the bloodstream. Here we've zoomed into the tissue of the digestive system, particularly the intestines. If we zoom in further, we'll be able to see an individual cell. Because glucose is a polar molecule with a molecular weight of 180 Daltons, it needs to use a transport protein to get into the cell. It's too large and charged to go through the hydrophobic tails of the cell membrane. Once the carbohydrate monomer enters the bloodstream, it's transported by the blood through the veins, capillaries, and arteries of the circulatory system. The carbohydrate monomer glucose will continue to travel around the circulatory system until it is absorbed by a cell somewhere in the body. The image here shows glucose being absorbed by a muscle cell in the leg. We've zoomed into the muscle tissue. If we zoom in further, we'll be able to see an individual cell. As discussed before, because glucose is over 100 Daltons in weight and is also polar, it requires the use of a transport protein to enter the cell. Once the carbohydrate monomer enters the muscle cell, it may be stored later for use as glycogen. Enzymes within the cell will assemble multiple glucose monomers to form a new carbohydrate polymer. This concludes our tour of the human body. Thanks for joining us.